Hello everyone, welcome to my channel GVS Chem. In this video, I am going to explain Biomolecules Part 3 Amino Acids and Proteins. Please subscribe to my channel, like, share and comment my videos. Now let us learn about amino acids. Substances containing both amine group and carboxylic group are called amino acids. Substances containing both the NH2 group and COOH group are called amino acids. So this is the example for this is the basic structure of amino acid here, amine group and here carboxy group. Depending on the position of the amine group with respect to carboxy group, amino acids are classified into alpha, beta and gamma amino acids. So here this is C, NH2, COOH, and of course here this is CH2, this is CH2, this is CH2, CH3. Now this is a functional carbon atom. Functional carbon. So with respect to functional carbon, this is alpha carbon, this is beta carbon, this is gamma carbon. Now in this particular substance, NH2 group is present on alpha carbon, therefore this is alpha amino acid. Alpha amino acid. If the amine group is present here, let us take this example. This is H, this is H, COOH, this is CH, and of course here NH2 here, this is hydrogen. So amine group is present on beta carbon, therefore this is a beta amino acid. Now amino acids have trivial names. Alpha amino acids, especially alpha amino acids have trivial names which are derived from the property of such amino acid or their source of availability. So depending on the property of the amino acid or depending on their source of availability, amino acids, alpha amino acids have trivial names or common names. For example, this is a very simple amino acid. Here this is C, NH2COOH. This is referred as glycine. And of course amino acids are represented by three-lettered symbol or one-letter symbol. So three-letter symbol is GLY and one letter is G. So this is the general formula of amino acid. This R, that is alkyl group, if it differs, if we take different groups in place of R, that is CH3, C2H5, etc., then we get the different substances. For example, if you take R equal to CH3, this is COOH, CNH2, hydrogen, this is CH3. Therefore, this is alanine. Three-letter representation is ALA, one letter is A. Right. Now, if C equal to CH3 taken twice CH, so this is COOH, CNH2, hydrogen here. This is CH, this is CH3, this is CH3. So this is isopropyl group, this group is, and this substance is known as valine. This substance is valine, V-A-L-I-N-E, valine. So three-letter representation is VAL, one-letter representation is V. So in this way, if R differs, then we get different amino acids. Now, depending on the requirement by the living body, amino acids are categorized into two different groups, essential amino acids and non-essential amino acids. So essential amino acids are those which are not synthesized in the living body. So in our living body, in the animal body or human beings, certain amino acids are synthesized in the body itself. Such amino acids are not necessary to supply through the diet. So such amino acids are referred as non-essential. Whereas amino acids which are not synthesized in the body, they are referred as they must be supplied through diet. Therefore, they are referred as essential amino acids. So essential amino acids are those which are not synthesized by the body and which should be necessarily to be supplied through diet. For example, valine, leucine, lysine, etc. All these are essential amino acids and they should be supplied through diet. On the other hand, non-essential amino acids are those which are synthesized in the living body and are not necessarily to be supplied through diet. That means these amino acids they are synthesized, they are obtained in the body itself. For example, glutamic acid, aspartic acid. Glutamic acid and aspartic acid, they are non-essential amino acids. So here, this is glutamic acid. This is aspartic acid. So this is the R. This is the R. This is R. This is glutamic acid. Of course, depending on the number of amino groups and carboxy groups, amino acids are classified into different categories. That is, this is, see here, in this one acid group is there, one more acid group is there. So here acid groups are 2, whereas basic group is only one, that is NH2 group. Here NH2 group. Therefore, this is referred as, since acid groups are more, this is referred as acidic amino acid. This is acidic amino acid. Whereas in this case, this is aspartic acid. In aspartic acid, acid groups, how many are there? Only one. Basic groups, base means amine groups. One is here, one is here. So basic groups 2 are there. Since 2 basic groups are there, this is referred as a basic amino acid. This is lysine is basic amino acid. So amino acids are classified into 3 categories as just now we discussed that. That is, first one is depending on their nature. The first one is neutral. Acidic amino acids are those relatively more number of carboxy groups than amino acids. So acidic amino acids have more number of carboxy groups than basic groups, that is amine groups. So basic amino acids are those having relatively more number of amino groups than carboxy groups. Amino acids are usually colorless, crystalline solids, soluble in water and behave like salts. In aqueous solution, carboxy group of amino acid lose a proton and amine group accepts that proton and hence form a charged species. So here this is the amino acid and here this proton is lost, therefore this becomes COO minus and this proton is accepted by this NH2 group, therefore this becomes NH3 plus. Therefore this species has both the charges, therefore this species has both the charges. Therefore, this species is known as Jotter ion, ZWITTER, Jotter ion. So Jotter ion has both the charges, positive charge as well as a negative charge. Now, let us see the amino acid once again. This is carbon, COOH, this is NH2, this is R, this is H. Now, in place of this R, if you keep hydrogen, this is, what is this substance? Glycine. Now, one more, in place of COOH, NH2, in place of R, let us keep CH3, therefore this is alanine. So in alanine, see here all 4 groups with respect to this carbon atom, all groups are different. Therefore this is, what is this carbon? This carbon is chiral carbon. Since it is chiral carbon, this alanine is optically active substance. So except glycine. Except glycine, all other amino acids are optically active substances. Therefore, they rotate the plane polarized light either to left side or right side. Amino acids are moderately soluble in water. We discussed that amino acids exist as a deuterion, that is, they have a positive charge as well as a negative charge. So, in acidic solutions, amino acids they exist as cations, and in basic solution, basic solution in basic medium, they exist as anion. At intermediate pH, they exist as dipolar ion. Dipolar ion is nothing but a deuterion, that is, they has a positive charge and negative charge, that is deuter ion, right? So such pH at which the deuterion exists is called isoelectric point, is known as isoelectric point. So this is isoelectric point is the pH where the amino acid exists as a deuterion, is known as a. So this is a amino acid COOH group. This is NH2. This is hydrogen. This is R in place of hydrogen. Therefore, this is glycine. Now, let us write one more amino acid. This is naturally occurring amino acid. So here, this is COOH. This is NH2. This is H. This is R. In place of R, let us keep CH3. So what is this? Alanine. Now let us compare this with glyceraldehyde. What is glyceraldehyde? We discussed already in carbohydrates. This is a CHO group. This is a OH. This is a hydrogen. So this group is CH2OH. Now for this glyceraldehyde, it was taken if a OH group is on right side, this is referred as a D-glyceraldehyde. So all the amino acids are compared with glyceraldehyde configuration. Therefore, depending on the position of the NH2 group, so in place of OH in glyceraldehyde, in amino acids NH2 group is there. So as the NH2 group is on right side, this is referred as a D amino acid. This is D amino acid, right? Therefore, here we have COOH. This is NH2. This is H. This is CH3. What is this substance? Alanine. Now another one is COOH. NH2 is on left side. This is hydrogen. This is CH3. So both are alanine. So with respect to glyceraldehyde, in this alanine, NH2 group is on right side. Therefore, this is D-alanine. This is L-alanine as NH2 group is on right side. Now, all these are naturally occurring amino acids. So, most of the naturally occurring amino acids, naturally occurring amino acids have L configuration. Most of the naturally occurring amino acids have a L configuration. Now let us take an amino acid. The basic amino acid is this is COOH. This is NH2 group. Hydrogen. What is this? This is glycine. Now let us take one more glycine. This is let us write the NH2 group here this side. Therefore, this is CH, CH. This is COOH. Now here, this OH group of carboxylic acid and hydrogen of amine group of OH group of one amino acid combines with the hydrogen of amine group of another amino acid. They combine. They are removed as a water. This is water. Therefore, what do we get here? This is nothing but this is H. This is hydrogen here, NH2. This is C double bond O, NH. Here, this is NH. So, this is CH2 COOH. So, what is this substance? What is this group? This group is nothing but this is amide group. Amide group. As this amide group is formed in between two amino acids, this is referred as peptide linkage. Peptide linkage. So this is peptide linkage. Now since this peptide linkage is formed by two amino acids, this is referred as a dipeptide. Dipeptide. What is dipeptide? Dipeptide means it is nothing but amide group formed in between two amino acids. If three amino acids combine, we call it as a tripeptide. Tripeptide. If four amino acids combine, we call it as a tetra. And that is if more and more number of amino acids combine to form peptide linkages, that is referred as a polypeptide. Polypeptide. Now keep this point in mind that two amino acids of same group, same variety are different amino acids. Same type of amino acids are different types of amino acids. They combine to form polypeptide linkages. So these polypeptide linkages are present in proteins. Proteins. Polypeptides having molecular mass more than 10,000 are referred to as proteins. Some polypeptides with fewer amino acids are also referred as proteins. See, in general, polypeptides with more than 10,000 molecular mass as referred as proteins, but in some cases, polypeptides with less than that number, that is fewer amino acids, are also referred as proteins. For example, insulin is a protein which controls the blood sugar. Is the protein of 51 amino acids. So proteins are of two types, that is fibrous proteins and globular proteins. Fibrous proteins are thread-like molecules lying side by side, held together by hydrogen bonds. So fibrous proteins are the main structural material of animal tissues. Generally, they are insoluble in water. For example, keratin in skin, hair, nail, wool, horn and feathers. So the keratin protein is present in all these substances: skin, hair, nail, wool, horn, feathers. Collagen is another protein present in tendons. Myosin is a protein present in muscles. Fibrin is a protein present in silk. So all these are the examples of fibrous proteins, and of course they are the structural material for tissues. Now globular proteins, they have molecules which are folded into spheroidal shape, that is a spherical shape. They are smaller and occupy less areas. So since they are spheroidal or spherical shape, they occupy the less areas, and molecular forces are generally weak and they are soluble in water. They perform a variety of biological functions. For example, insulin and thyroglobin, they catalyze the biological reactions. Some globular proteins, they regulate the biological reactions. For example, insulin controls the blood sugar. Some globular proteins act as antibodies. Antibodies means they protect the body from ailments like fever, etc. Structure and shape of proteins are studied in 4 levels. Those 4 levels are primary, secondary, tertiary and quaternary structures. Now let us discuss about each and every structure here. Now the primary structure means proteins may have one or more polypeptide linkages. Each polypeptide linkage has some amino acids, and these amino acids are joined with one another in a definite sequence. This sequence of arrangement of amino acids is referred as a primary structure. So the sequence is definite. If the sequence changes, then it becomes another protein, that is a different protein. Now the secondary structure refers to the shape in which the polypeptide chain exists. Secondary structure exists in two different shapes, that is alpha helix structure and beta plated sheet structure. Now let us discuss these different structures of secondary structure. So here the most important one is alpha helix. So this is alpha helix structure. Alpha helix structure. So alpha helix structure is the most common ways in which a polypeptide chain exists from all possible hydrogen bonds. So hydrogen bonds are possible in this. So since hydrogen bonds, so here these are the hydrogen bonds. So because of these hydrogen bonds, from one amino acid to another amino acid in the next step, therefore this alpha helix looks like a twisted chain. So this is a twisted chain. On the other hand, now the next one is a beta sheeted plate structure. So this is beta plated sheet structure. Beta plated sheet structure. So in beta plated sheet structure, all peptide chains are stretched out to nearly maximum extension and then laid side by side, which are held together by intermolecular hydrogen bonds. So whether it is alpha helix structure or beta plated sheet structure, the most important point one has to notice here is there exists a hydrogen bond between amino acids, different amino acids present in the protein. So because of that, because of the presence of hydrogen bonds, the different structures are obtained. The structure resembles the plated folds of a draper, and therefore it is known as a beta plated sheet structure. Now let us discuss the tertiary structure of proteins. Tertiary structure of proteins is nothing but the overall folding, nothing but the overall folding of proteins. What does it mean? The secondary structure further folds to get the second tertiary structure. So in tertiary structure, there are two types of proteins are obtained. Two types of proteins. What are those? Fibrous proteins and globular proteins. Already we discussed about this in the beginning of this structures. So tertiary structure of proteins is nothing but the overall folding of the proteins, that is secondary structure further folds to get a tertiary structure. The result of tertiary structure gives two types of proteins, that is fibrous and globular. Now let us see the quaternary structure. Quaternary structure means some proteins may have more than one polypeptide linkage. So when the protein has more than one polypeptide linkage, the spatial arrangement of all these, the spatial arrangement of different polypeptides of a protein is known as quaternary linkage or quaternary structure. quaternary structure. Now, proteins found in biological system. Proteins found in biological system with three-dimensional structure and biological activity. Every protein has its own biological activity. So, proteins found in biological system, that is living bodies, with the three-dimensional structure and biological activity are called native proteins. So, when proteins are subjected to protein is subjected to physical change. Physical change means change in temperature or chemical change. Chemical change means change in pH. So when temperature changes or pH changes, then what happens? Protein loses its biological act. Protein loses its biological activity as the structure disturbs and hydrogen bonds break, and this is called the loss of biological activity of protein. The loss of biological activity of protein due to physical or chemical changes, physical or chemical change is called denaturation. So the loss of biological activity of protein due to physical or chemical change is called denaturation. The best example is boiling of egg. So during boiling of egg and curdling of milk,